been eating, but not necessarily in that order. So I'd like to try and bring up our first speaker so we can start right on the dot, and that's Peter Treblin. Is that said right? Tiblin? Yeah, Tiblin. And he's going to be talking about pike, and his title is Be on Time or Be Dead. So that's a good title to start with.
All right, so thank you for the introduction. So everyone here is on time, so that's good. So what's behind this curious title? There's one who's not on time. So I have worked with the spawning migration of pike. So they live in the Baltic Sea and they migrate to small streams for reproduction every spring. And this species is interesting to use to study variation in migratory timing. So it's a huge predatory fish, the one you see here. That's more from my interest as a sport fisherman. But it's a long-lived species, so it's up to 25 years old. It's heteroparous, so it's a multi-event spawner, meaning that we can actually get long-term longitudinal data for behavior. And they display homing behavior. So they return to the same streams every spring. So using migratory capture methods, we can actually shed new lights on the causes and consequences of individual variation in timing. And that's both among individuals as well as within individuals. So animal migration, all of you know about it, is, at least from what I see, one of the most fascinating phenomena in nature. So it's widespread in many different kinds of organisms. And it's, I would say, well understood that timing is important when you move between habitats. But the understanding of such movements and the causes and consequences thereof is mainly based on studies on birds. So for the other kinds of organisms, especially fish, there are not much done on this kind of questions. And that's the scope of the study that I here present. So related to the causes and consequences of timing, I asked 4 different questions. So the first one is whether individuals are consistent in timing. Do they arrive, is a variation within individuals less than variation among individuals? Secondly, I was interested in whether the timing of individuals are influenced by experience. So do they change behavior when they become older? And moving on to the consequences, I wanted to see whether we could see fitness consequences of arrival timing. And having access to data that are longitudinal, I was also interested in to see whether we can assess the long-standing issue on whether flexibility is adaptive. So, all right, so here is the wonderful pike again. And for you who doesn't know, this is a widely distributed top predator. It's found throughout the northern hemisphere. It's found in predominantly fresh waters, but also in brackish waters. It is suggested to be a keystone species. And I work in the brackish water area of the Baltic Sea. So that's a big red dot there in the middle. And here we have the migrating pike, and I will just go through this life cycle quite fast. So they spend about 97% of the life cycle in the Baltic Sea. When it's time for reproduction, which occurs in April, May, they migrate to those small streams. And that migration is not like bird migrations from Africa to the Arctic, but they are about 5 to 10 kilometers generally, but they can be up to 100. So this is the kind of system we have. So what we did here is that, you see, those streams are pretty small. They are about 3 meters wide and a couple of decimeters deep. So we shut them off completely with those pike nets, meaning that we can actually sample the full cohort of migrating individuals. And by marking those individuals, we can generate recapture histories. So we sample this stream for 6 years, and we follow more than 2,000 individuals. And we follow whether they come back or not. So the ones here represents that they are alive, and the zeros that they are dead. So every one here, we have a date. And that date is actually, we use standardized dates. So the population in itself shifts the timing a little bit between years depending on the onset of spring. So what the results I will show you is based on standardized timing. All right, so 2,000 individuals, 6 years of monitoring. What did we learn then? So first of all, are individuals consistent? So what we do here is that we're comparing variation within an individuals to variation among. So it's a measure of repeatability. And yes, they are consistent. And what I want to show here, I will show you schematic figures. If you want to see the original figures, they are in a paper published earlier this year in Animal Ecology. So what I want you to see here is that we have different individuals, and although that they vary in timing a little bit, that variation is less than variation among individuals. So you could say that they consistently arrive early or late, year in and year out. So next, we wanted to test whether we could see a pattern in arrival timing across years within individuals. So once again, we use longitudinal data, and we look at how the timing changes across years. And what we found is that it seems like those pike are fine-tuning the timing. And what do I mean with that? So this is one of the original figures. So here on the y-axis, we have magnitude of adjustment between years. And here we have the years. That means the adjustment in days between year 1 and 2, 2 and 3, and so on. And what I want you to see here is that, and all the lines are individuals, that the adjustment becomes less and less when they become older. So they get more and more precise in the timing, suggesting that they actually are learning from their earlier mistakes, so to say. So they're using the experience to get more precise in their arrival to the streams. And this finding is, as far as I know, pretty new for fish at least. So, moving over to the consequences of timing. So first of all, being an heterospecific species, surviving between years is extremely important for fitness. But it's also that we didn't know whether migrating to the stream was associated with survival. So first of all, we looked at whether variation among individuals in timing influenced the survival. And yes, it did. Hence the weird title, Be on Time or Be Dead. So what we found is a collinear relationship between timing and survival. So first of all, that tells us that the arrival timing do matters for those adult pike. And if you want to be too general about it, it seems like being too early or too late will impair your survival. So, all right, so that's one too much. From that on, we went on to the intriguing question, whether it's an adaptive value of being flexible. So what we did here is that we worked with the magnitude of adjustment within individuals to see how that influenced survival. So you see those arrows here, there are differences in magnitude between individuals, how flexible they are. And we found evidence that being flexible actually influences your longevity. So here we have two individuals. You see the yellowish one here and the blue. And if you look at the yellow one, he or she is very flexible across for the first 2 years of migration. So when they start migration, which is about the age of 2 to 3 when they become mature, if you change a lot the first 2 years, you will live longer. And although that's not a causal link in any way, it's still suggesting that it is an adaptive value of having a capacity to be flexible. So that's all. What I want you to remember is that we have studied individual variation in timing for a fish, one of the first studies doing so. We studied this wonderful species pike in the Baltic Sea that migrates to those small streams. And we monitored the timing across 6 years for 2,000 individuals. We found that individuals are relatively consistent in the timing, and they also have the ability to fine-tune the timing with increased experience. Moreover, the arrival timing is important for survival, so it influences fitness of the individuals, and also that having the ability to be flexible, or be flexible, have a positive impact on your survival. So with that, I would like to thank my co-authors, Per Larsson, Anders Forsman, and Tobias Borger, and co-workers, and also the funding people. And that's all. Thank you.
So being on time, actually under time, so we've got some time for questions. Any questions, please? Yes.
I just wondered what it was about timing that mattered. So being early and being late makes a difference in your survival.
Yeah.
So what is it that was causing that cut-off?
We don't know that for sure, but we have two explanations. The first one is that arriving early, since they switch habitat, they will be exposed to different pathogens. And after the winter, the immune system of fish are generally quite low. So that could be a reason of why being too early is bad. Being late, then you face all the other pikes that are spent fish, and they are hungry. And pike is highly cannibalistic, so that would probably be associated with predation.
I was wondering the pattern that you showed with the flexibility in timing of young, and that seems contradictory to the survival pattern that you had with the early and late. I mean, when they are so flexible when they are young, they must have kind of a lower survival because they are probably deviating from the optimal maximum survival.
Yeah, I agree. So, I mean, if too early, too late is bad for you, then it should not be good to be flexible about it. But maybe, so if two fish arrive really early, and one is capable of change that, whether the other one is more strict, then that probably will be an favorable trait. So I agree that it's a bit contradictory, but yeah.
I was just wondering if the arriving early or late was something that was heritable.
Now, we don't know about that. Depending on the consistency, the repeatability, maybe it could be. And yeah.
Is there any evidence that if they get it right in their first year, in the sense that they arrive at the right time and therefore they have to change their behaviour less, so they have to be less plastic, is there any adaptive benefit to that?
I mean, that should be the best strategy. But in our data, we have not seen it.
All right, I think we'll let you off. So if any more questions, I'm sure people will take them at lunchtime or
Yeah.
during the next couple of days. And so thank you very much.
Thank you.
We'll start on half-past, so you have got about a minute or so respite while we change. Okay, right. I just shut that down.
Yeah, sure.
Can go between rooms. So
Yeah.
some people might be coming and going. This is move out and try and get to the next room. So.
They're running already.
Feel free to chat amongst yourselves, get to know some people, just like silence. Okay, so I'd like to introduce our next speaker in the Behavioural Ecology session, and it's Miguel Gomez, and he's going to tell us about relaxation in sexual conflicts.
So my talk is about how ecological factors help to shape the dynamics of sexual conflict. In specific, how heterospecific competition can reduce harassment over females and increase their survival. Okay, so sexual conflict has been broadly defined as the differences in the evolutionary interests of individuals of both sexes. And theoretical models predict that it can lead to a perpetual coevolution in which the females are evolving traits or behaviors to decrease the mating rate, while the males are continuously evolving to increase it. However, most of the work done so far has been on the lab or in very simple ecological conditions, and we don't know a lot about how interspecific interactions can help to shape this conflict. So what we did is we used this species of damselflies, is Calopteryx splendens. On your left side is the male. He's characterized to have a patch of color in the wing, and the size of the patch is correlated to the fitness. So the larger the patch, the fitter the animal. And on your right side is the female, is less conspicuous and has almost no pigmentation. And in the northern part of the distribution, in Finland and in Sweden, where we did this work, they can coexist with this cogeneric species, is Calopteryx virgo. So this is a bit larger species, is more aggressive, and is dominant over splendens. And it's characterized to have this, the full wing, the whole wing, full of melanin. And our hypothesis is, as these species, the males compete for access to the females, and Virgo is dominant over splendens, they can outcompete them and kind of exclude them from the territories, and that creates kind of a safe area for females where they're not harassed as much. Okay, and the first question we ask is if the presence of the heterospecific has an influence on the survival of the female. So we made this experiment in which we set up these large outdoor cages, and each cage has water containers and vegetation to simulate natural conditions. And we put damselflies there, controlling for the species composition. So in some cages we have only splendens animals, that was our allopatric treatment, and in some others we have both species, and that's our sympatric treatments. And we also control for the sex ratio, so some of them they were even sex ratio or skewed towards males. And we color mark each animal. So each animal has an individual tree color code, and that allows us to monitor the survival of each animal in each cage on a daily basis. Okay, so we found that the presence of the heterospecific does has an influence on the female survival. In the y-axis we have the female longevity, measured in days, and the x-axis are species composition, so sympatric or allopatric treatment. And in the skew sex ratio, you can see that it seems to be a difference between allopatric and sympatric. Like, it seems like females do better in the allopatric treatments. However, it wasn't statistically different. It looks like it is, but this is because we have the random effect of the cage. What was significant was in the even sex ratio, the light blue color. The females do much better in the sympatric treatments than in the allopatric treatments. And well, as I stated before, this is because of the interference competition, which Virgo is dominant over splendens, and they outcompete them. And that reduces harassment of the females. So that's the next question we ask, right? If the heterospecifics interfere with splendens mating attempts, and this is reducing the harassment. So we did a second experiment using these IKEA laundry baskets, because it was Sweden, so you have to go local. And we put damselflies there, controlling for the species composition and the sex ratio in a similar way as in the outdoor cages. But in this case, we color dust mark the males. So we apply color dust in these two regions. So this is the clasper. It's in the tip of the tail and in the genital area of the male. And when you see the reproductive behavior, first the male clasps the female by the thorax, and then the female has to bend her abdomen to reach the genitalia of the male. So after 24 hours in these laundry baskets, we can just search for traces of the color dust in these two areas of the female, and that will tell us if the female mated or not. And as each male has a different color, we can tell which male mated which female, and related to morphometric measurements. So that's what we did, and we found that mating success of the males is affected by the presence of heterospecifics. So in this case, we have the male mating success in the y-axis and the size of the forewing length in the female. That's a proxy, very good proxy for the female size. And you can see in the light blue color, that is the allopatric treatment, that males are quite successful mating the females, and they are more successful the larger the female. But in the sympatric treatment, we have this nice relation in which the mating success of the male, it's less likely they have less success mating with the larger females. So we believe that this is because of the presence interference competition of the Virgo. So Virgo, as I told you before, is a larger species. Females are also larger, and they identify these large splendid females as conspecifics, so they guard them against mating, and that's why we see this decay in male mating success. And we did check for heterospecifics matings. So this is Virgo males trying to mate or mating with splendid females, and it was on 50% of the females from the sympatric treatments, obviously, and it was all with these large females. I think the smallest one was 34.2 millimeters, something like that. So it kind of points out in this direction of that misidentify the species, and that's why they are guarding them. Okay, but is this harassment affecting the survival of the females? So in this, in this species, mating is quite costly. The males chase the females, then they clasp them by the thorax, and the female tries to force her way out, and then they mate. But in successive matings, the males can remove the sperm from previous matings. So this means it's not only harmful for the female, but it also means that she has no benefits from repeated matings. Not even genetic benefits of multiple parenting. It's just, there's no trade-off between cost and benefits, it's just the costs. So if it's true that there's interference competition with the large females, then we should see an increased survival of the large females, but only in the sympatric treatments. And that's what we found. So this is, again, the longevity of the females measured in days and the proxy for the female size. And yeah, the light blue color is the allopatric treatments, and the survival of the female is quite uniform in all the sizes. But in the sympatric treatment is dark color. The survival increases with size, which kind of indicates and points out in the direction of our hypothesis of interference competition. Okay, so just to conclude, right ahead of time, I think, is that, yeah, I think we have good evidence to show that the community composition does affect sexual conflict. And we believe that studies on sexual conflict, and especially theoretical models studying speciation under these dynamics, should include more ecological realism, because, as we show, interspecific interactions can modify the strength and the direction of the conflict, and that can prevent or increase the evolution of reproductive areas to emerge. However, this is just kind of half story. We still have to measure, and we hope that we can do it in the next field season, the other part, from the male perspective, so the male aggression and the effect of male density in this conflict. So yeah, Justin Tucker is my supervisor in Manchester, Eric from Lund, Hannah helped us in the field, and yeah, NERC is the funding agency.
The speakers have been all under time. They're incredibly efficient today. So we've got plenty of time for questions on this one. Yes?
Next to talk, is it known what sort of signals are being used by the dragonflies to identify each other as the same or different species?
Not completely. So we know that the females are very good about identifying their own males, and we believe that is because of the color patch. the size of the patch or having the full wing full of melanin. But I know there's some studies trying to find out, apparently they have good indicators that there's pheromones involved, but the males have the pheromones. So when the male claps the female, she can identify the species by the smell. But we know from previous studies that Virgo is not very good at identifying their own species because they're kind of the dominant, so they don't really care too much.
Did you guess any of the Splendens males try to make the smaller Virgo females? So did it work the other way around?
We didn't work with the Virgo females. They're kind of harder to find. We didn't have enough sample size to work with them.
How good are the females at rejecting males? So once these dominant products come in, can they get away?
I haven't seen one that can break away, but they do evolve kind of a resistance to mating. So they have to actively bend their abdomen to reach the genitalia of the male, and they cannot do it if they don't want. However, if the male is too persistent, they just simply do it and get away with it. Yes, but very low. Apparently I only know from one study that has done it, and it was kind of less than 30%, but the survival of the 30% of matings, but the survival of the offspring wasn't like good at all. It was like less than 1%, something like that. So they try, but the offspring is not viable.
So do these two species obviously coexist, and what is the natural sort of community composition? Does it naturally occur, this dominance?
Yeah, so normally Virgo is distributed from, that's like where they coexist is the southern part of the distribution, and the Splendens is the opposite. That's the limit of the northern part. And they're kind of dominant, but they're ecologically very similar. So we are still wondering how are they able to coexist in very stable, because they have been coexisting since the last glaciation, basically, 10,000 years ago. And we don't really know how.
Another question to add to your list.
Yep. Some work to do.
Okay, so we'll let you go. Thank you very much, Miguel. You have a few minutes before the next talk if you want to chat or if you want to go to the toilet or between sessions. I'll just go on this. Okay, so time for our next speaker, and this is Jim Vavadis, and he's going to be talking about food-mediated climate impacts. And I love your identification of birds there. That's like my identification skills. A small brown wetland bird.
That's what it is, yeah. Thanks very much. Yes, I'd like to talk to you today about how climate is seemingly benefiting a small brown wetland bird. But given this is a behavioural session, what I really want to talk to you about is how food supplementation studies, despite their biases and despite their weaknesses, actually can provide some very interesting insights to the impacts of climate change on wild populations, like small brown wetland birds. So, what other small brown wetland bird could I be talking about, other than the ultimate small brown little bird, which is the Eurasian reed warbler? It is not much to look at. It's not exactly the pinnacle of natural beauty. It is no Belize jaguar, but it's obviously doing something right. It is ubiquitous across Europe and the low swamplands of the UK. It is abundant. It is increasing, and it's expanding its range. So what do we know about the ecology of this species? Well, we know that it is a very skillful and agile predator of aerial invertebrates. And this differs in that respect to the wetland cousin of the reed warbler, the sedge warbler, which we know through behavioural and molecular studies is less agile and less efficient predator and instead relies on slow-moving species or slow-moving prey. So, for example, slow-moving aerial predators at the beginning of the day where the temperatures are cooler, less able to catch them mid-flight, for example, like a reed warbler might. So this big difference between these two species of wetlands might underpin the big differences between their habitat selection and the wintering grounds. So, for example, reed warblers are able to occupy the so-called suboptimal habitats in the Sahelian region, so the scrublands, whereas the sedge warblers are confined to the wetlands, so where the prey is much more abundant, so they are confined to these wetland habitats. And as a consequence of that, they are actually more prone and more vulnerable to the density-dependent mortality often seen in wetland habitats. So during the Sahel drought periods, the wetlands contract, and sedge warblers, like many of the other wetland species, actually undergo this big density-dependent population crashes. So this is actually one of the big reasons we have this uncertain population trend in sedge warblers. But this is all a bit of an aside, really, because reed warblers aren't declining. They're not beholden to this impact. So why are reed warblers actually increasing as a result of climate change? And one of the big reasons is, on the breeding grounds, warmer temperatures are actually driving invertebrate abundance higher earlier in the season. Pretty simple, the warmer it is, the more invertebrates there'll be. Pretty straightforward, really, relationship. So what does that mean in reality? So when the reed warblers arrive at the breeding grounds, they will lay their clutch when the invertebrates are at very low abundance, and by the time those eggs hatch, the abundance will have increased. They will then feed the chicks, and at this point the invertebrate abundance will be at a certain level where you can see why they might be tempted to have a second clutch. But we can see through long-term studies in the European populations, the second clutch success rates are quite low, between 0 and 15%. So what we're seeing now is the trend of warmer temperatures earlier in the year is actually increasing and widening this food peak, so that as well as breeding slightly sooner, the success of the second broods is actually increasing up to 30%. So this alone could account for this trend of increasing reed warbler population trend. I've been studying reed warblers probably for the last 7 years or so, and been conducting food supplementation studies with them, and I've probably fed a few hundred of them. Mealworms, some fed, some unfed. And I've measured consistently, so this is feeding them at the point of arrival. So I feed them before they start breeding, and I've measured consistently that some of the ways that they actually respond to feeding in the fed populations, that they consistently will increase their incubation mass, they will advance their breeding date, they will reduce the duration of incubation, they'll fledge their chicks sooner, and they'll have reduced predation. And this is all good and well, very interesting insight perhaps, but unfortunately, these, apart from the predation, these are all hallmarks of supplementation studies. This is what happens if you feed birds more mealworms, they're going to get fat. In fact, a lot of studies show that if you actually are feeding terrestrial vertebrates more food, they're going to increase their body mass, they're going to breed sooner, they're going to reduce their home range. So actually, what is this telling us? What are the insights that we can gain from it? So despite it not necessarily being an accurate representation of the future, particularly under a climate change scenario where we don't know how much invertebrate abundance is going to change, we can gain some insights from these food supplementation studies. For example, by even accepting food during the breeding season shows that they are to some extent food limited. You try feeding a reed warbler any food in the Sahel, in the so-called suboptimal habitats, and they couldn't be less interested. So by even accepting food, it shows they are to some extent food limited. Being able to change their breeding date, so by advancing their breeding date, gives us the insight that perhaps they are evolved to breed within a very competitive environment. So in mainland Europe, they have to compete with much more superior species, wetland species like great reed warblers and Savvy's warblers. So they must fit in their breeding around these more competitive species. So if they're given the opportunity to breed sooner, with more invertebrate availability, they'll do it. Incubation, we know that incubation is actually determined by the amount of food available. If there's more food, the adult birds have less reason to leave the nest and leave the eggs. And if we're actually encouraging them to breed sooner, during perhaps the colder time of the spring, then they're even less likely to leave the eggs. So actually, incubation duration isn't necessarily telling us very much here. But let's focus on predation and parasitism. In songbirds, the biggest driver of recruitment and productivity is actually to do with nestling survival, and this starts at the egg stage. So with reed warblers, the biggest sources of nest failure are to do with weather, so really poor weather will cause nest failure, predation by sweeter predators, and parasitism by cuckoos. So in typical reed warbler populations, nest success or nest failure is actually about 30%. In our population, sometimes it can be 50%, so a really bad storm year, 50% nest failure. In our population, it was about 30%. In our fed population, it was less than 5%. So it's a big reduction in predation. In our South Wales population, we've got no cuckoos in full-time employment there. So this is all to do with the predation of the chicks by moor hens and by magpies and by jays. So the big question is why is this happening? What is the big deal with predation and them changing it with increased food supplementation? Is it by decreasing the incubation period? Are we actually reducing the period of vulnerability? By breeding slightly earlier in the year, are we actually avoiding the peak predation period? Or are they staying near the nest? If you're going to give them food nearby, are they going to be able to react quickly? Do they simply have more energy to react to predators? Or are they presenting fewer predatory cues? So if they're not leaving the nest as much, they're not displaying where the nest is. So we set out to test this in 2 studies. So we did an incubation attendance study. In the far picture there, you can see that's a reed warbler nest before any eggs are laid in it. And there's a little cable you can see just draped around the reed going to a data logger. So we can see where the bird is on the nest or not. And then we did a predator presentation study where we presented a predator to see how quickly the birds would respond. So we found that in the incubation attendance study, birds were consistently coming back sooner. So in the maximum off-bout duration was less than a minute, so a minute sooner in the fed population versus the control population. And even in the mean off-bout duration, so per trip, it was about 7 seconds shorter. So it's 300 trips or so per day. So that wasn't changing. So they're still making these trips, but they were just shorter. In the predator reaction study, we showed that in the absence of a predator, we sort of see a similar relationship. So the fed birds are coming back a little bit sooner, but not very much. But if you put a predator in there, they'll zip back straight away. You can see the big difference between the predator and the control and the fed population responses. So again, what does this really tell us? This is food supplementation. How much are we really gaining from this? Well, we know that fed birds staying close to the nest, they're able to react when they need to react. So that's one insight. Interestingly, there was no aggression to the predator mount. So either that's because they weren't convinced that it was a real threat, very bad taxidermic mount perhaps, or it could be that it's not really, the reaction isn't to do with nest defense at all, it's to do with vigilance. Remember, this is a species which is one of the biggest hosts for cuckoos. So if they are vigilant about anything that's in their vicinity of their nest, they can be more wary about any changes in their clutch, and they can reject any additional eggs. So very interesting insight about defense versus vigilance. So in conclusion, food supplementation studies may not be accurate reflections of future scenarios unless you're exactly replicating what you predict will be a change in the amount of food available to these very agile and skillful predators. So it's important to understand their limitations. That's the first point. But they can reveal very important insights about how your species is responding. So for example, reed warblers are very responsive to food. And lots of their breeding parameters are potentially optimized. How much of that you should accept is questionable. But the most convincing of these is the nest vigilance, which is a result potentially of cuckoo parasitism rather than nest defense. But if you can achieve 2 benefits, being wary of the cuckoo's trickery and deterring predators, then it's a pretty watertight adaptation. And that's it. So work is ongoing, but I'd like to thank my collaborators and support. Thank you very much.
I was going to ask you what your predatory mount was, but I can see it.
Yeah, it's a
now.
Stokes. Yeah, sorry. Yeah. Stanley, that is.
Okay, any questions, please? Stunning silence. That's down to me now. Okay, so I was thinking about your supplementary feeding and what are the other consequences of potentially supplementary feeding in terms of maybe the behaviour of the predators, that they could learn where the birds are, you increase numbers, increase aggregation?
Yeah, that's right. That's true. Yeah. So one of the side effects of supplementary feeding is that you're going to get a lot more birds in the area taking advantage of the food. So that's another really big limitation of supplementation. So, for example, blue tits will find them very, very quickly, and they will compete with the reed warblers. So one of the things that we actually have to do, including predators coming in like crows and magpies, is actually to protect the supplementation station. So there's an area where they would come for food, and we'd actually weigh the birds as they were getting the food anyway. So we're getting this mass data, but we'd create a cage over the station so that only reed warblers and small birds could get in, but it deterred a lot of more wary species to go into this cage, whereas reed warblers quite happily to go in and come out again. So it was a really good solution to that.
So can I pick up on that? So why would you potentially use supplementary feeding to boost population but not in long term? And you are going to have these community effects. So, you know, what is the point and
what we're trying to understand here is why this species is increasing. It's unique in the sense that it's one of these species which is actually benefiting from climate change. So it's picking out where the benefit is coming from. So it's not coming from necessarily the wintering grounds where they're avoiding that the mortality of the effects of drought is actually happening on the breeding grounds. So by simulating an increase in food, we can say this is probably where this increase is happening. And so pinpointing the mechanisms of population increase. Yeah.
Thank you, Jim. If anyone does have any questions on something, we'll take them during lunch or coffee or any time catching this week. Okay.
Okay.
Thank you very much. Just give you a second. Yeah, so that one is the leader and that forward. Okay, so our next speaker is Elizabeth Morgan, and she's going to be talking about European shags and consistency in foraging behaviour.
Yes. Yeah, so I'm talking about essentially repeatability, and I'll start with a bit of background on why repeatability could be important to ecology. And when I say repeatability, I'm meaning consistent individual differences in behaviour that are retained for a given timescale. And there's a growing body of work on various taxa that have showed repeatable behaviour in a wide range of species. And this is important because if individuals aren't ecologically equivalent, then they might not be responding to changes in their environment in the same way. So those organisms with greater flexibility are going to find it easier to adapt to changes. And it's going to be especially important in behaviours that could affect fitness. For example, foraging behaviour. And my little example here shows what might happen if there was a sex difference in the area's species of foraging, and we just took into consideration as a whole what we know, and didn't consider that certain species, certain individuals are just using one area. If something changed in that area, it would then have the potential to skew a population and ultimately have population level effects. And we want to look at this in the European shag. So we know that repeatability in foraging behaviour exists outside of factors such as sex in seabirds. But so far, most of this work has focused on long to mid-ranging species, things like gannets and albatrosses that go hundreds or thousands of miles from their breeding sites to feed their chicks. We want to know whether the same is true for short-ranging species. Now, shags are very, very short-ranging. Sort of 3 kilometres is the average trip distance. So they could virtually see where other birds are foraging, and it would logically be less energetically expensive than to switch their foraging areas. However, they are a benthic diving species, so they forage on or near the seabed. So having prior knowledge of where the good areas to find food are would benefit them. Plus, they make about 3 or 4 trips a day, so they're ideal candidates to look at repeatability on because we can get a lot of trips over a short time scale. So what I'm going to be talking about today is I'm going to ask a few questions. So are shags consistent in how and where they're foraging? To see if these levels of consistency are retained both within one season and between multiple breeding seasons. To ask whether individuals express different levels of repeatability in their foraging tactics, and ultimately ask, is there any fitness consequences to this? So we've been working on the Farne Islands in Northumberland on a population of shags just off the Northumberland coast. We've been deploying GPS and depth recorders on chick-rearing adults over the last 3 breeding seasons. From this telemetry data, which is about 3 or 4 days worth of data, we extract foraging trips and assign behaviours based on speed and diving data. This gives about 3 or 5 days data. On an average, we've got about 8 trips per bird. And in our data set, there's about 52 individuals, and some of those individuals we have multiple years data for. And this is just to show you, oh crap, sorry. I've got presenter view on. So this is just to show you where they're actually foraging. And in 2014 was a really good year, and they were foraging over a much shorter range than they were in 2016, this year, which was particularly poor. So are they repeatable? We used a similar parameter to I think we've already heard about today, which takes the variation between and within groups and gives you a repeatability score, where 1 is highly repeatable and 0 is not very repeatable. And we tested this on various foraging parameters, and the repeatable parameters were things like foraging longitude, departure angle, latitude, and also dive depth. And these things can be considered locational, because if you remember, I said they're benthic diving species, so there is a locational aspect to depth as well. Now that tells us there is variation in the population, there is repeatability in the population, but it doesn't tell us how each individual is expressing that variation. So do each individual birds have their preferred foraging spots? So to look at this spatially, we got kernels. We did kernel density for the first 8 trips birds, which made 8 trips made, and we used an affinity score, Bhattacharya's affinity, to give us a measure of overlap of how consistent in space these kernels are. So 0 would be low spatial consistency, and 1 would be complete spatial consistency. We averaged those for each bird and give them an average repeatability score. These were then ranked. And on the left here shows our most repeatable bird in 2014. You can see it's over the period of 3 or 4 days, it's going to the exact same spaces. I should mention that the smoothing parameter we used for our kernels was based on the area, the average area bird forages over within 1 trip. And you see WRP here is foraging in a variety of areas, but there is still a degree of overlap, and we created a null model for our overlap by randomizing individual identity, and all the birds within the population were more repeatable than if they were just foraging at random. So that's within a season. What about between seasons? So for birds we had multiple years data with, we did exactly the same thing, except instead of trips we used years. And these results show the birds that we have 3 years data for, and you can see largely they're foraging in similar areas from 1 year to the next. And this individual down here is particularly interesting because it's the one that was the lowest ranked repeatable bird in 2014, but you can see it's using those same areas again year after year. So most birds seem to be more repeatable between years than they did within a year. But ultimately, does this all matter? Are there any links between repeatability and reproductive success? So we used the within-season repeatability score for each individual as a measure of consistency in models. And to look at reproductive success, we used productivity, so the number of chicks that were fledged from a nest. And we also wanted to look at adult body conditions, so we used an index of mass over wing length. And we also wanted to look at another sort of measure of condition. We've just heard that birds in better condition may breed earlier, so we wanted to look at this. We used hatching date, and we standard hatching date between years to account for environmental variability. And we found that it does seem to matter for females. Repeatability was retained in most of our models, and there was a significantly positive relationship between female condition and repeatability. We also found a similar relationship in hatching date. So females that were more repeatable were laying earlier, and we think this is linked to bird quality, with the earlier birds being the better quality birds. We didn't find any effect of condition in males across the season, and we also didn't find any effects of, we didn't find any effects in males, which could be quite logical because it's the females that are producing the eggs, so their condition is more reliant on their breeding dates. We didn't find anything for productivity. We couldn't find an effect of more repeatable birds being more productive. So to conclude, as a population, Farne Island shags are repeatable in most locational trip parameters. Most shags don't seem to be changing their foraging spots. However, there is variation in individual spatial consistency both within and between years. Some birds do seem to be more repeatable than others, and this seems to matter if you're a female. So we did find links between repeatability of timing and breeding condition, and we think this is more of an intrinsic factor rather than environmental, because over the same time scale, we didn't find that effect in males. So it's potentially due to bird experience and quality, and this fits with some previous work that's been done that showed older, more experienced breeders that breed earlier are more effective at providing food for their chicks. And we're arguing that potentially this is because they know exactly where to go to find that food. So the implications of this would all depend on how fixed or flexible these sort of best females are at breeding. If prey distributions change, are they going to be able to adapt and move, or are they set in their ways and are going to be unable to adapt? And I'm afraid I don't have the answer to that question yet. So this is just a few thanks to you for listening, and to everybody that's helped with my project so far. Thank you. Yeah, so so shags do seem to learn from their parents. They will hang around with them after fledging, and there's big flocks. So there does seem, whether it's their parents or whether it's just sort of social learning, of the best areas to go. But it's been shown that as they get older, they become more efficient in finding food. So yeah, I think it's their learnt ability, but I think they learn it early on, and whether these females that are repeatable also know other locations, and if one location's gone, they've got that knowledge there from previous years that they'd be able to switch or not. I don't know.
Are you aware of any personality studies in shags that might combine, for example, sociability of shags, that some might avoid high densities, and that's why they're potentially using other foraging grounds?
I'm not aware of any sort of personality studies. There is a study that says that they're foraging socially in some areas, but I haven't seen that in my population, so I don't know whether it's a widespread thing or whether it's an environmental, it depends on the environment that they're foraging in. So yeah, the body condition was the mass that they were when we caught them. So it would have been after they laid their eggs because we were catching birds on young chicks. So.
Is it possible that your hatching date drives your consistency in that if the prey is more reliable or that you get hotspots of productivity early in the season that you might get more consistency as opposed to the other way around?
Possibly, but because we didn't see in the males over the same time scale, we think that there is something in the condition as well, rather than just sort of an environmental decline in productivity, because the males that we were tracking at the same time haven't shown this decrease in repeatability across the season.
Any questions? Again, I'm sure listeners will take questions outside the meeting, so thank you very much for a really interesting talk. We do just have a minute or so to wait for the next talk, so again, please chat and sound, make some friends. Be social.
Tell everybody per Twitter what a great session we are having here. What they're missing.
Can I check with you how you say your surname?
Huh?
Can I check with you how you say your surname?
Gregor?
No,
My surname? Oh, Kalinkat.
Kalinkat.
Yeah.
Kalinkat.
That's great.
I'm dyslexic. All my letters jumble up. It's really quite annoying. I learn it and then I have to relearn it again. Okay, so we'll go on to our next speaker, Gregor Kalinkat, and he's going to keep with the theme of variation in feeding, but also in other types of behaviour as well.
Thank you for the introduction. Thank you everybody for being here. I have a bit of cough and sorry if I yeah. So what you see here in this picture is one of Europe's biggest lignite power plants, and it sets the stage for the system we are studying, and I want to present a little bit of data that we gathered this year with organisms from there. So this is in Western Germany, and this is where the heated cooling water from the power plant comes out. And this whole area here is heavily influenced by anthropogenic activities. Large-scale open-cast mining has totally modified also the thermal regime and the running of the Erft River, in which our little model system, the Gilbach, mouths here, which later mouths into the Rhine over there. And so this is not only a very heated river, but we have a lot of non-native species. That at the very mouth, where you just seen the picture. So you do find some native fish there. This is a chub. And then we have guppies, thousands of them, and established population for at least 40 years now. So this is like 1 kilometer downstream, even more guppies. And yeah, these fish are basically dumped there by aquarium pet owners, and you have like a whole bunch of aquarium species. So this is the main actor today, the convict cichlid, which forms part of the data that I will later present. So you might ask, why should we study such an artificial system where like very artificial temperature regimes that have nothing to do with the real world outside? And on the other hand, the tropical species that won't probably tolerate and won't become the temperatures in Central Europe and become really invasive here. But I think it's quite interesting to study, because what you see here is a very brand new theory for thermal tolerance curves in general thermal biology that just came out like 2 weeks ago. And you see here that the scope for the cold adapted species is much broader, because the slope for the metabolic relation with temperature is shallower here than it is over there. And so this is what they state, like, because the slope is much steeper here, the thermal tolerance window for species with tropical origin is much smaller. So what happens in the Gilbach and in other artificial systems where you have these high temperatures and a lot of non-native species is that you have, on the one hand, pushing the native species into this direction via the thermal pollution. This is the direct effect, but generally we have the global warming effects that affect the system. And on the other hand, you have these species introductions with species that are often more tolerant towards warmer temperatures than the native species. And on top of that, which is like what we find in the Gilbach, but has also been shown for other systems, is that there's a pre-selection process happening in the aquarium trade. Just by transferring species and breeding them, you select for cold tolerant species. So this is data from the guppies from the Gilbach, and these two are, so these are the guppies that have been living for many generations in the Gilbach, and that are just your guppies from your pet store downtown, and those are compared to wild types in Venezuela. So they are also pre-selected to tolerate cooler temperatures via the processes in the breeding industry. And then the second question is, to set the stage for what we're looking at, why do we want to look at personality traits and like repeatable behavior when we look at species introductions and species invasions? Well, basically individual level variation matters a lot as the driving force for evolution, which is quite clear, I guess, for everybody in the room, but also for non-linear ecological processes like feeding relationships, consumer resource feeding relationships dependent on resource density. So this has been gotten quite some attention during the last couple of years, and this book just came out like 1 month ago, but there's also like this call for looking at how specific personality or behavioral traits are associated with species that are like really competitive and invasive and even harmful in many places in the world. So I have might be add that like the species that we see here in the Gilbach, they might not be a real threat in terms of invasiveness in Europe, but they are like really typical invasive species in other parts of the tropics. So They come from the cichlid, for example. They come from Central America, but they have been problematic in Australia, for example, and also in other parts of the world. So we look at how these traits depend or correlate with dispersal tendency and invasion success. And also, like more general, as I said, that the nonlinear relationships are important, and the interspecific variation has to be taken into account. And so, what we want to look here at, like, how do these relations between behavioral traits and the success of the introduced species work along a temperature gradient, coming back to the Payne and Smith study mentioned before, and also looking at how this connects to higher order ecological functions that happen between, like, not only within species, but also between, for example, consumers and resources. And also in invasion ecology, there has been a lot of studies, like very recently, applying the function response approach, so resource density dependent feeding rates to assess the impact of invasive species. So another thing is, like, as I mentioned before, that individual variation is, or also as others have mentioned before, that it's in many parts it's not really accounted for. In functional response studies, generally the way is to describe them at the population level. So if you do it like in experiments, you have a lab population of 30, 40, 50 individuals, and you have, like, each data point that you need for determining your functional response is like one individual feeding one time. So in a study that was published last summer, we took this one step further towards the approach that was presented here, and measured functional responses. So each individual was presented with varying prey densities over a set of replicates, and this is what we found there in this particular study with this least killifish, it's called, and the Randria famosa. We did not find that the personality traits, so activity and boldness, are strongly correlated with the feeding rate of the individuals. We had a clear effect of body size, but we also have in this experiment we also had a wide a large range of body sizes that we used. So in this case, no personality effect. So now comes some data from the convict cichlids, Amatitlania nicofasciata, and we took them to the lab. Basically, they're juveniles, and we measured in. So this is the body sizes this time. Sorry, this time not such a big variation between 26 and 35 millimeters, and we measured them 6 times at 2 different temperatures, 23 and 29 degree, and checked in the same treatment before the feeding experiment, checked their activity levels and their shyness responses. So this is the overall shyness here. So what we found is that both traits are correlated, and at both temperatures, and that the shyness measure is repeatable. It's not a very high repeatable, it's around 0.3, but the activity measure is not at all repeatable. These are the functional response curves plotted for all individuals as one curve. So we had a problem that we couldn't fit the individual model that I've shown before from the previous study, because at the high temperature treatments, many of the treatments were not satiated. So the problem with individual level studies is that you can't do it like 2 months after, because like we usually do this with population level, you can just put something and add it later on, but with the individual level study, you just have to work with what you have. So you see here that there is a clear temperature effect, but it's not. I think it's not as pronounced as you would expect for a fish that comes from the tropics. So the final question is then, do personality traits in this case explain individual variation in functional responses? And here we have, like, just to compare, on the left-hand side, you have the shyness response, which is the repeatable of the two behavioral traits that we measured, and the residuals from the functional response model fitting. And you see there is no clear pattern, neither for the 23-degree treatment nor for the 29-degree treatment. But I mean, it's a work in progress, and maybe we find more here. So we found that, like, analyzing the overall data set, that there is a very significant effect actually of the temperature on the feeding rate, but also despite the very low variation in body sizes, that we have also a small but significant effect of body size on the outcome in the functional response. That's it?
Yep.
Okay, that's fine.
Sorry, you were going into your questions out. I've got time for one question very quickly. And we've got very little time, so thank you. If you have any questions, that's okay. That's all right. We'll go on to the next speaker, please.
Oh, it's come up, that's fine. I'll just do, hopefully, 5. Right, it's not. Can she come in? I just want to get him to so it's full screen. I can't see how you did on there.
Sticking with themes of predictability and foraging behaviour, and she's going to be talking to us about environmental predictability and whether it plays a role in shaping individual foraging.
Hi, thank you. Yeah, so I'm going to be talking about some work from the first year of my PhD on the role of the environment in shaping foraging behaviour. And within ecology, foraging behaviour is important for niche specialisation, which in turn is important for generating and maintaining biodiversity, both through facilitating speciation and also promoting coexistence. Niche specialisation is driven by competition and also by ecological opportunity. However, what can really underpin specialisation at the niche level is specialisations in individuals and populations. So individual specialisations in habitat use are widely documented across the animal kingdom. However, these vary in strength and magnitude between individuals and species. And these drivers of behaviour and their variation remain largely unknown. They may be both intrinsic, as Liz was mentioning earlier, but also extrinsic to do with resource availability, and it's the latter of these two that I'm going to be focusing on today. So as we all know, resources are fundamental for growth and reproduction, and as such, they are a source of both competition and opportunity, both of which drive specializations. And because specializations emerge through consistent behavior over time, this leads me to my study question, which is whether resources that are predictable in time lead to specializations in behavior. However, within the marine environment, these resources are very difficult to directly measure, but we can use physical parameters as a proxy for their distribution and availability. For example, depth and currents can lead to regions of upwelling and increased productivity. As you can see from the right-hand graph, the shape of the phytoplankton bloom in turquoise is influenced by currents which can shape their spatial distribution. So this leads us to a slight tweak of the question, which is whether a predictable environment leads to specializations. In order to ask this question, I'll be looking at three physical parameters, and for those of you in the audience less familiar with oceanography, I'll just run through them briefly and why they're important. Firstly, depth, which is static over time, this doesn't change, and this can drive water currents and also the structure of the water column. Tidal stratification is a measure of water column structure, and this can influence the amount of nutrients that are available within the photic zone where there's sufficient light for primary production, and this can in turn affect productivity. This is a really sweeping generalization, but you can imagine in a mixed water column, resources are distributed throughout the depth range, and therefore there's less nutrient and primary production held within that photic zone. Whereas in a stratified water column, where you have a sufficient density gradient to create two distinct layers, there's very little mixing between the two layers, and as such, the phytoplankton and the nutrients are held up within the photic zone, enhancing productivity. And lastly, fronts, and these are dynamic, they change in space and time, and these are where these two types of water bodies can meet. These can lead to upwelling of nutrients and therefore enhance productivity, and as you can see from the picture, they can be very pronounced at the ocean surface. So in order to compare across a range of predictability, we looked at two study sites. Firstly, Skomer Island off southwest Wales and Puffin Island off northwest Wales. Just to run through these briefly, at Skomer Island, the area in the vicinity of this site is very vertically stratified, and it has stronger fronts, therefore we might presume is more productive. And in all three physical parameters, it's much more spatially heterogeneous. And because of these three factors, we believe that there will likely be more predictable resource hotspots within the vicinity. In contrast, at Puffin Island, the water column is much more vertically mixed. It's much weaker in terms of fronts, and it's spatially homogeneous, therefore leading to much more unpredictable and uniform distribution of resources. So at our two study sites, we tracked the foraging movements of seabirds, and these are a model study species for behaviour studies. They're colonial breeders and as such experience high levels of competition, which can lead to niche segregation. So we studied the black-legged kittiwake, pictured top right, with GPS loggers, and the goal of tracking was to achieve multiple trips per individual, which we achieved at Puffin Island over 6 years and Skomer Island just during 2016. And these two right-hand plots, in case you're unfamiliar with tracking data, show one individual at each of the two colonies, tracked over multiple foraging trips in different colours. So the next step is to quantify resource selection by the population, and we start with our environmental gradient across the whole area, from which we can extract information for the environment that was used by the population from all of the GPS points. We can compare this in a resource selection model to the available habitat from a randomly generated set of points in a binomial mixed-phase model. So to move on to some results, these are density curves across the environmental range, in this case depth on the x-axis, for the predictable colony, Skomer Island on the top, and the unpredictable colony, Puffin Island on the bottom. And the dotted line shows the available habitat. This is from the randomly generated points. When we compare this to the used habitat from the GPS points in the solid line, you can see that at both colonies, they preferentially visit areas of shallower waters. And this is reflected in the output of the model. This is a probability curve on the y-axis from 0 to 1 across the environmental range on the x-axis for Skomer Island in the solid purple line and Puffin Island in the dashed line. And the important thing from this graph is that the slope is steeper at Puffin Island, indicating a stronger selection for this static feature at the unpredictable environment. To move on to stratification, again you can see differences between the used habitat and the available habitat. However, this time they're more pronounced at Puffin Island. Again they're more pronounced at Puffin Island. So again, the output from the model, the slope was insignificant at Skomer Island, whereas the steeper slope at Puffin Island again indicates that there is stronger selection for this static feature at the unpredictable environment. This time for fronts, and you can see that the difference between the available habitat and the used habitat is actually more pronounced at Skomer Island, whereas at Puffin Island the distribution of both types of habitat are much more similar. And again, back to the resource selection output, and this time the steeper slope at Skomer Island indicates that there is a stronger selection for this dynamic feature at the predictable environment. So just to recap on these three results, both of the colonies selected for shallower depths, however this was more pronounced at Puffin Island. The selection for stratification varied. It was very weak at Skomer Island, whereas again this was more pronounced at Puffin Island. And however for fronts, both colonies selected for weaker fronts. I'll come back to this, but this time the selection was stronger at the predictable environment. So what can we conclude from this? Well, in a heterogeneous environment where we are likely to get more predictable hotspots and much more dynamic features, the population was more specialized on these dynamic features. In contrast, at the homogeneous environment where we're likely to have a much more unpredictable resource distribution, the populations were specialized on static features in the environment. And this perhaps is driven by this opportunity to specialize. In the absence of dynamic features, the population is relying on static parameters. And this is likely to have consequences under environmental change. Perhaps if a population is adapted to static features, it's therefore less able to respond to environmental changes in regime. I mentioned that both populations selected for weaker fronts, and this is perhaps counterintuitive since I said right at the beginning also that fronts are likely to be more productive. I think that actually there's a lot more complex things going on, and one thing I'm going to move on to looking at next is different measures of frontal activity, including persistence and so on. And lastly, I mentioned right at the beginning that individual specializations are really important. All of these models are quite preliminary, and there's some early evidence to suggest that there's individual variation in resource selection, so there's a significant effect of individual in the model. So my next question will be whether environmental predictability influences the strength of individual specializations between the two colonies. And I'd like to just say thanks to everyone that helps in the field, and thank you for listening. Yeah, so I think there's definitely differences in the availability. At Skomer Island there's much more stratification, so this might lead to an increase in production. It's quite hard to measure these things comprehensively and directly, so that's why I've focused on the predictability, but something to look at would definitely be like the chlorophyll layers. Having said that, there's a bit of decoupling between the primary production and the seabirds because there's trophic levels in between them, so I think it's certainly something to look at with perhaps additional study sites across a gradient of predictability where there might be differences in productivity and try and untangle those.
I mean, it sounds like it could be that you see differences because the total productivity is different between those two sites. Maybe if there's more food in general, you can afford to chase the dynamic things, whereas if there's less food, you have to go where you know it's going. I mean, is that wrong? Is that a possible interpretation? Or?
Yeah, I guess so, and something that I've not necessarily thought too much about, but I think since the stratification and the depth still can influence productivity. And I'm maybe just getting confused in myself. But yeah, I think it would be interesting.
But just to follow up on that, perhaps one way to disentangle those two possibilities of overall abundance and predictability would be to expand the study to incorporate tracking from other sites by other groups, no doubt. And it would allow you to generalise to a good degree.
Yeah, we're hoping to get some kitty weight tracking data from other colonies. There's lots of places where this work's going on. So if anyone has any of that data, then please let me know.
Sometimes I also find myself in a difficult situation that you have to set the available resources to some space. You've chosen to give the same environmental space to both islands, so to speak. Do you have a good reason for doing so, rather than so the background, the availability, is limited in this square?
Oh, it's actually limited by the extent of the colony's foraging range, so it is slightly different. Sorry, I didn't explain that. But yeah, it's different between the two. It's just easier to show us.
Yeah, maybe you slightly covered it just there, but I was just interested in like the spatial scale at which you determined your kind of stable environment versus unpredictable, and whether like it kind of is important that the spatial scale at which the species forages over as well, and whether if you increase the spatial scale of your environment, whether at different scales you will see different levels of predictability depending on the different environmental features.
So I quantified this predictability within the foraging range of that exact colony, but that is different between Scone Island and Puffin Island. And at Scone Island, they actually travelled a lot further. They went on much longer foraging trips, and I'm not really sure if we can explain that in itself. But if you expand the measure of predictability wider at the two colonies, I'm quite certain that it would be quite similar, because the environment around both the colonies is still quite similar, and they're both quite different from each other. Yeah.
Okay, thank you very much, Alice. I think we've interrogated you enough. I think there's some key people here that you're going to have some really good discussions with over coffee to help inform the next year of study as well. So good luck with the next year. Okay, thank you. So we're on to the last speaker of our session before...
Oh, can I ask you to turn off the presenter view?
Yeah, do you want to click it?
We're on to the last presenter of this morning/afternoon session just before lunch. And I'd like to introduce Samantha Patrick. And she's going to be talking about...
I think I've broken it. I don't...
She's broken...
Oh, no. Is it? No, it's all right. It's on there.
It's okay. It's going through.
It's okay there,
yes,
just not
I
here.
can see it on there.
Okay.
Are you okay?
Yeah.
Okay. Samantha Patrick, and she's going to be talking about boldness and the exploration trade-off.
Okay, so one of the main questions that we face in ecology is how do individuals find food? And one of the reasons that this is complicated is because the distribution of prey is very rarely uniform. And instead we find that it's normally something more like this, where it's clumped in the environment. And this leads to two specific questions I'm going to look at today. The first one is, how much time do you spend in a patch? So once you find one of these patches, how much do you exploit these resources? And then when do you leave this patch and search for other areas and explore? And the prediction is that when the quality of a specific patch drops below a certain level, that this threshold should be the moment when you should move and then look for new resources. But what I'm going to talk about today is what if this threshold differs between individuals. So one of the reasons that you might get differences in this threshold is there might be differences in the risk or the payoff of different strategies. So if you imagine an individual that leaves a patch sooner, it might then find higher quality resources somewhere else, but in addition, it might then not find these resources. So in contrast, you might get individuals staying in a patch for longer, and this will enable them to exploit the resources they know are there, but it will limit their ability to find better quality patches. And the things that we think might be drivers of these differences are things like risk-taking, which might be driven by intrinsic variation. So what are the possible intrinsic drivers? The first might be age. So we know that as individuals age, their survival probability decreases. And with this, you get a prediction that they'll increase their risk-taking to invest more heavily in current reproduction. Another factor might be individuals' personality. So we know that bolder individuals are more risk-taking. And so for this population here, there might be differences in prey choice as well. And what we know from previous research is that these differences are predominantly driven by sex. So as you can see from the map here, we have really strong sexual segregation, and you get females traveling to the north of the colony and males traveling to the south of the colony. And the consequences of this is that you get differences in temperature. So as you move further south, you get decreases in temperature and increases in productivity. And this then leads to the hypothesis that the environment will drive differences, and that males will have higher exploitation because there's higher productivity where they forage, whereas females will favour exploration because there's lower productivity. And finally, these strategies might exist if these fall along a trade-off. So an example of a trade-off might be the difference between the number of patches and the time an individual spends in the patch. So an individual that spends a long time in a patch might end up with fewer patches, but individuals that spend short times in the patches might end up with many patches. And we propose that these might be alternative stable strategies in our population. So to look at this, we created a framework so we could test our hypotheses, and we examined the behaviour along a continuum of exploration to exploitation. So firstly, we predicted that it was going to link to personality and to boldness. And there were two main reasons for this. The first is that we know that bolder individuals are faster explorers, and they tend to explore novel environments, whereas the slow explorers, they tend to be slightly reticent to explore, and therefore we thought that they would favour exploitation of known patches. We also know that bolder individuals are more risk-taking, and this would encourage them to favour exploration, whereas shy individuals are risk-averse, and therefore they should favour exploitation. As I just outlined, we think that older individuals will take the more risky strategy of exploration, whereas younger individuals that have a higher survival probability will favour exploitation. And finally, specific to this population, we predict that females will favour exploration due to lower productivity in the environment, whereas males will favour exploitation because there's more prey available. We also link this to specific foraging traits to allow us to examine whether there is a trade-off between exploration and exploitation. So we predicted that exploration will be correlated with less time in a patch, whereas exploitation individuals will spend longer. Exploration will be associated with smaller patches, and as a consequence, potentially, we predict that individuals will have more foraging patches if they favour exploration. And finally, we think that individuals favouring exploration will have fewer foraging attempts or lower foraging effort compared to those individuals which exploit the patch more thoroughly, who exhibit more foraging attempts. So this leads to three questions that I'm going to try and answer during this talk. The first is there a trade-off between exploration and exploitation, and does this result in a strategy that we're able to detect? Second, are these alternative stable strategies in the population where you find no fitness consequences? And finally, what are the intrinsic drivers of these differences? So I work here in the Crozet Archipelago in the southern Indian Ocean, and I work on a population of wandering albatrosses that have been studied since the 1960s. And as a result, we have age and sex estimates for all of our birds. So in 2009, we started a personality project on these birds, and we tested their boldness in response to a human. So we have about 1,600 tests. We have 1,600 individuals tested and about 3,500 tests. So it's a very simple score. We simply approach the bird, and we record their response from a score to 0 to 4. So a bird that has a low response would have 0. If it moves its head, it would have a score of 1. If it raises onto its tarsus, a score of 2. If it vocalizes, a score of 3. And if it stands up, a score of 4. And what this represents is it represents a continuum from shy to bold, with the shyest individuals displaying no response. And we've previously shown that this score is both repeatable and heritable in our population. So then to look at foraging behaviour, we used GPS loggers. So we used off-the-shelf loggers that we then removed the housing and waterproofed. We caught the birds and then taped the loggers onto their back. We then let the bird go to sea, and then when the bird came back to the nest, we caught the bird and recovered the loggers. And we have data for about 200 individuals, and there's 172 of known boldness. So how do we assess exploration and exploitation? So from the GPS tracking data, we isolated areas of intense search using area-restricted search. And these are areas where the bird slows down and increases turning. And we considered these to be areas of exploitation. We then isolated the travel between these patches and considered this to be exploration. And as a result, we were able to estimate the size of the patch, the time in patch, the number of foraging patches, and the foraging effort. So what did we find? The first question was, is there a trade-off between exploration and exploitation? And what we found was strong evidence to support this. So there was a positive correlation between time in patch and foraging effort, such that birds that spent longer in a patch had higher foraging effort. These birds were also associated with larger foraging patches, and these individuals had fewer patches. And we found the same relationship for foraging effort and size of patch and the number of patches. So yes, there does seem to be a trade-off, and this seems to be associated at one end, the exploitation end, with a long time in the patch, high effort, large patches, but few patches. We then looked at a principal component analysis to try and identify whether we could categorize an exploration-exploitation strategy. And we were able to do that, and the principal component had a positive weighting for time in patch, foraging effort, and size of patch, and a negative weighting for the number of patches. So this again represents an individual that favors exploitation, and they spend a long time in the patch with high effort, large patches, but few patches. So then what are the fitness consequences of these strategies? So we looked at the reproductive success in the year of tracking, and we found no evidence of fitness consequences. So we looked at both the exploration-exploitation strategy, but also all of the variables individually. So we then looked at the intrinsic drivers of these differences. So when we looked at sex effect, we found very few variables that correlated. We did find, however, that males had fewer patches than females, but this didn't correlate with any other aspects of the trade-off. And when we looked at age effects, we found that there were no effects of age on any of our variables. So then we looked at boldness, and we found very strong effects of boldness on exploration and exploitation. So on the y-axis here, you have exploration at the bottom and exploitation at the top. And on the x-axis, you have shy individuals on the left and bold individuals on the right. And what you can see is that shy individuals strongly favor exploitation, whereas bold individuals strongly favor exploration. And when we looked at individual factors as well, we found that when you look at the size of patch, it's the shy individuals that have large foraging areas, whereas the bold individuals have small patches. And when you look at foraging effort, again you find that it's the shy individuals that have high foraging effort and the bold individuals that have low foraging effort. So we find strong evidence that boldness does predict an individual's position along this trade-off, suggesting that bolder individuals favor exploration, they have smaller foraging patches, and they have lower foraging effort per patch. So what can we take from this? What are our conclusions? So I think there's strong evidence that there is a trade-off between exploration and exploitation. And what we seem to suggest is that this leads to alternative stable strategies in the population, where we don't have any fitness consequences. So why do these differences exist? Firstly, we find no effect of age, which is interesting because we thought we might get shifts. And this suggests that perhaps the strategies are fixed in individuals, such that if personality is so important, individual strategies don't change with age. But to look at this thoroughly, we need to have longitudinal data to see whether individuals consistently use the same strategy over time, and therefore, as they age, are still using the same strategy as when they were young. We find little evidence of sex differences, except for the fact that males have fewer patches. But this is not part of the trade-off, so this is not correlated with any changes in other traits. So what this might be explained by is if males are making lower foraging efforts. They have all the same traits as females, but they make lower foraging effort per patch. Or equally, it might be that males have higher success per patch. So they make fewer foraging attempts simply because they're more successful for each foraging attempt. And so other intrinsic drivers, we showed that boldness strongly predicts an individual's strategy. And we think this might be due to intrinsic differences in exploration or the risk-taking propensity of bolder birds. So the next thing I want to look at is whether individual quality affects the strength of the trade-off. So there's a prediction that you might get weaker trade-offs in high-quality individuals, as they might be able to optimize all components of the trade-off. And finally, this provides a mechanism by which you might get differences in foraging behavior. And previous studies have shown that single foraging traits are not associated with fitness consequences, and this might simply be because they're part of a trade-off, and the full trade-off is not being captured. And finally, we've revealed individual thresholds linked to marginal value theorem, predicting when individuals should switch patches. And so we suggest that future work should try and model individual level variation when considering these models. Thank you, and here's some shameless advertising. Oh no. I'm not sure I understand what you mean. Yep. It's because of a small sample size. So essentially, we do have a slight skew in our data such that you have more shy birds in the population than bold birds. And there's some evidence that birds become shyer with age, and we think that's why there's a skew. So I think in young birds, you get a really nice normal distribution, and then you get some shifts with age. It's also much harder to track birds that are really bold. So if you go up to a bird and it tries to kill you, you're quite unlikely to put a logger on it. And so we do have a skew in sample sizes for that reason. Yeah, so we know that there's an interaction between age and personality on where birds forage. So bolder birds always travel further as they get old. So the females go further north and the males go further south. So that's why we fitted age in the models, because we know that they do have these age-specific foraging grounds, but that still doesn't seem to reveal any differences. So we did think that as birds make these longer foraging trips, as you say, they might find more occasional prey that's less clumped. The next step is to look at whether individuals actively forage on the shelf versus pelagic waters, so looking at the actual depth at which they forage, whether that's associated with differences in the actual foraging trade-off.
So we've run out of time. Obviously, everyone's very hungry for lunch. I'd just like to thank all the speakers for this morning for amazing diversity of talks, lots